Good evening, students. So welcome to Unacademy EAMCET YouTube channel. So myself Dr. Rajendra Prasad. So today's class will be discussing about the lesson that is cell the unit of life. So in that we will be discussing about the prokaryotic cells. In last previous classes we were discussing about the size and shape of the cells. So in this class we will discuss about the prokaryotic cell. So before starting the class, so good afternoon all of you. So welcome to So once again, good evening students. So good evening to Vishnath, good evening to Someswar Rao. So before starting the lesson, I would like to introduce about our Unacademy Plus subscription as usual, students. So Unacademy is providing daily. YouTube classes. So Unacademy is providing daily YouTube classes, students. So daily at 4 p.m. we will be having the biology class. So daily Unacademy is providing YouTube classes. So at 4 p.m. we will be having the biology class, students. So today my biology class, in that we will be discussing about the prokaryotic cells. So before starting the class, so I would like to introduce about our Unacademy Plus subscription. So there is a comprehensive batch is started. So in that comprehensive batch, so I will be taking the biology classes and Dev Prasad sir will be taking the physics classes, and Sachina sir will be taking the chemistry classes, students. So in this comprehensive batch, so complete syllabus will be covered, and tips and tricks will be given by the educator, students, and topic wise practice papers. So topic wise practice papers also will be provided. So and full length mock tests also. So full length mock tests also will be conducted. So once again, so good evening students. So good evening all of you. So good evening Sanjana. So with this comprehensive batch, so if you are interested, you can take 1 month plan that is costing 2,250 rupees, students. So there is a long term plans also available. So long term plan is of about 10,125 rupees. So and a 12 months plan. So 12 months plan is also available. It is costing about 12,375 rupees, students. So what is the importance of this Unacademy Plus subscription? So Unacademy's Plus subscription is, so premium content. So premium content will be available at affordable price, and a structured live course and dedicated live courses, dedicated live document sessions will be taken. So quizzes also will be conducted at regular intervals. So a batch course. So batch for complete preparation. So batch course also will be completely preparation. So and test series. Test series also will be conducted, students. So all this you can get for, so you can get for single subscription of 6 months plan of 10,125 rupees and 12 months plan of 12,375 rupees. So welcome to few more students. So welcome to Banu Prakash. So good evening, Banu Prakash. So okay, so let us continue with our topic in today's class. So once again, good evening, students. So in today's class, we will be discussing about the prokaryotic cell. So I will be discussing about the topic that is called as a prokaryotic cell. So according to Whittaker classification, so Whittaker has given 5 kingdom classification. So Whittaker has given 5 kingdom classification. In that Monera, so monerans, so these prokaryotic organisms are placed in a kingdom that is called as a Monera. So especially see these monerans, so in 5 kingdom classification of Whittaker, so prokaryotic organisms like bacteria are placed under the kingdom. So Monera kingdom, it is placed under the kingdom that is Monera. So this kingdom Monera contains the solo organism. So that solo organism is nothing but it is bacteria. So this bacteria is also again 2 types like Eubacteria and Archaebacteria. Eubacteria is the true bacteria, whereas Archaebacteria is the ancient bacteria. So that was given. So bacteria is, this Monera is further classified into Eubacteria and Archaebacteria by the Carl Woese, and he has given his 6 kingdom classification. So like that, so in the classification, if you see the classification of prokaryotic cells, so prokaryotic cells are placed under the kingdom Monera, and further this Monera is further classified into again Eubacteria as well as Archaebacteria by Carl Woese, and he given as the name of the kingdom as 6 kingdom classification. Anyhow, so what you have to remember is, so prokaryotic cell, so this prokaryotic cell is placed under the kingdom, so that is a place in a kingdom that is called as a Monera. So this Monera includes, so what type of organisms are included under Monera means, if you see Monera includes the unicellular, so it includes unicellular prokaryotes, no doubt about it. So Monera includes unicellular prokaryotes. So Monera includes unicellular prokaryotes. So unicellular prokaryotes are all placed under this Monera actually. Okay, so let us see about this particular prokaryotic cell in detail. So no doubt there is one and one solo organism. So there is one and one only solo, okay, of a single organism. So that is the bacteria. So bacteria is present, is a part of the, is the best example of prokaryotic cell. Okay, so let us see about the details about this bacteria cell in detail. So Sai Murthy was asking me one question. So he is telling that he got marks of 63 marks. So I think 63 marks, right? So 63 marks based upon your handicap quota. So okay, so you got 45% handicap quota. So I think you can have a chance. You may get a chance in you may get a chance in agriculture, no doubt about it. You have 45% quota, so you can get chance. So Murthy, I think you can get a chance. No need to worry about it. So better you should have written ICAR exam also. So anyhow, so let us start, students. So bacteria are unicellular organisms, no doubt about it. Bacteria unicellular organisms. So no doubt they have single cell. So bacteria unicellular organism. So that is a single cell. So unicellular organism means it is a single cell. Single cell is performing all the activities. It is a single cell. So single cell is performing all the activities. Such type of cells are called as a unicellular. So we already have seen previously, so bacteria is the best example of unicellular organism. Apart from bacteria, some protistans are also there. They are also unicellular in nature. So this bacteria unicellular, no doubt about it. So bacteria was first discovered. So who has discovered bacteria? First discovered. So bacteria was first discovered by Anton von Leeuwenhoek. So Anton von Leeuwenhoek is the person who has first discovered bacteria. So from where he has discovered? So he has discovered from the rainwater, actually means pond water, means the rainwater which has stagnated in the pond. So that water is called as a pond water actually, that is a stagnant water or rainwater. So from the rainwater he has discovered bacteria, and also from the scum of the teeth. So what is the scum of the teeth? So early morning. So early morning when you wake up from the bed, so you just do some scratching on your teeth. Then what happens? You find some substance is, the sticky like substance is present. That is nothing but it contains the bacteria, and that is the scum of the teeth. So Antonov Leeuwenhoek has discovered, no doubt he discovered bacteria. He discovered bacteria 1676. So very long back actually. So where he has discovered, he discovered in the pond water and as well in the scum of the teeth. Okay, so that is the thing which we have to remember here. So bacteria mostly are pathogenic. So majority of members of bacteria are pathogenic to humans. So what is the meaning of this term pathogenic means? So these bacteria causes diseases. So the bacteria causes diseases. That is the reason they are called as a pathogenic organisms. So bacteria which causes diseases, those are called as pathogenic. So apart from the bacteria which is causing pathogen, which is causing diseases to human beings, there are some bacteria which are also called normal flora. So they are present on our skin. So everywhere on our body there are some thousands of bacteria, lakhs of bacteria present, but they don't cause infections. Okay, but there are some other bacteria which causes infections actually. So anyhow, so bacteria are mostly pathogenic on human beings. So where bacteria are found in almost all places. So if you see where bacteria is found, so bacteria is present in almost all places. Almost all places means where can we see bacteria? Bacteria can be seen in the air, bacteria can be seen in the soil, bacteria can be found in the water. So that such type of condition is called ubiquitous. So there is one specific term is used. So bacteria is present almost in all places. There is a specific term called ubiquitous. So what is ubiquitous? Bacteria is distributed in all places. Such condition is called as ubiquitous. So the bacteria is found in almost all places. So there are some rare places where humans also cannot stay, like hot springs. Even in hot springs also bacteria stays. So if you see the temperature in the hot springs, the temperature in the hot springs generally ranges between 80 to 90 degrees centigrade. Okay, so that much high temperatures also this bacteria is surviving. So not all bacteria, but some specific bacteria. So they can also survive in the very high temperatures, and such areas where high temperatures are present, they are called hot springs. So even bacteria can survive in icebergs. So in icebergs, if you see the temperature of icebergs, certainly it will be less than 0 degrees centigrade. So even in case of the low temperature, so less than 0 degrees centigrade. So less than 0 degrees centigrade is present in the icebergs. So in that regions also this bacteria grows. So apart from that, the bacteria also growing in the intestine of the man. So it is also growing in the intestine of the man. So they are growing in the deep in the soil and in the seawater. So they in different areas. So bacteria is growing in almost all places. It is in different places. It is growing in hot springs. So they are growing in the icebergs, intestine of the man, and deep in the soil. So different places. So bacteria is staying alive. So once again, good evening students. So good evening Chetana. So good evening Ashwadhama. So Ashwadhama, you can crack NEET first rank, no doubt about it, but you have to do your hard work. So teacher role is of just only 20 to 30%. So to get better rank, it is the student's role to get the better rank. So student has to nearly 80 to 70 to 80% hard work should be done by student itself. A teacher's role is only 20 to 30% in a student's achievement. So that is the main thing which you have to remember. So teacher can guide you, but getting rank means you have to work hard. Certainly you will get it, no doubt about it, if you do with dedication. Okay, so thank you Asudama. Okay, so let us see students. So let us see about the shape and size of the prokaryotic cells. So if you see the prokaryotic cells, so prokaryotic cells vary greatly in size, no doubt about it, they greatly vary in size. So if you see the size of the prokaryotic cell, so size of the prokaryotic cell may range from 1 to 10 microns. So generally this bacteria, so in typical bacteria, if you see in our book they write typical bacteria. So typical bacteria size ranges. So typical bacteria size ranges between 3 microns to the 5 microns, typical bacteria. So like that anyhow, so these prokaryotic cells like bacteria, they may range size up to 1 to 10 microns, no doubt about it. Okay, so in that specifically smallest bacteria is called Dilister. So Dilister pneumocinitis. So it is the smallest one. So you can see it is ranging between It is ranging between the 0.1 to 0.3 microns. In our NCERT textbook they write smallest bacteria is nothing but it is a smallest bacteria. If you see in our books they write like PPLO, pleuropneumonia like organisms, so PPLO. So that PPLO also belongs to, so this dilister only. So PPLO is also belongs to dilister only. Actually the PPLO is also belongs to this dilister group only. Okay, then so largest bacteria, if you see largest bacteria, so largest bacteria is Spirillum volutans. So Spirillum volutans. So PPLO size is 0.1 microns. PPLO means pleuropneumonia like organisms. PPLO. So what is this PPLO? Pleuropneumonia like. So pleuropneumonia like organism. So PPLO, that is the smallest pneumonia like organism. It is of 0.1 microns in size. Okay. So it is clearly given in NCERT textbook. So dilister also is a member, belongs to this pleuropneumonia like organisms only. Okay. So largest bacteria, no doubt largest bacteria is Spirillum volutans. So largest bacterium is also Spirillum volutans. So welcome to Pawan. So good evening Pawan. Welcome to Unacademy EAMCET YouTube channel. So if you see the shape of the bacteria varies in difference. So the bacteria shape varies. Okay, so bacteria greatly varies shape. So 4 basic shapes of bacteria are there. If you see, the first one is called coccus, second one is called as a bacillus shape. So third one is called vibrio, it is also called as a comma shape. Spirillum, it is called as a spiral shape. So 4 basic shapes of bacteria, that is coccus shape. So bacillus, it is called as a rod shape. So vibrio is called comma shape, and spirillum is called as a spiral shape. You can see this diagram. So this is the coccus shape. So this shape of bacteria, coccus shape bacteria, you can see there is flagella is not present, flagella absent actually in this bacteria. So flagella absent. So in coccus shaped bacteria, most important point you have to remember is in coccus shaped bacteria, so flagella will not be present. So in bacteria, like in a rod shaped bacteria or spiral shaped or comma shaped bacteria, so you may expect flagella may be present in some members, but flagella is totally absent. Flagella is totally absent in the coccus shaped bacteria. Okay, so that is important thing which you have to remember. So rod shaped bacteria, no doubt best example is bacillus. You can see the bacillus. So bacillus, this bacteria is also forming spores. So this is sporulating bacteria, bacillus. So spirillum, so this is the spirillum shape. It is spirillum shape. So it is also having flagella. You can see tuft of flagella. You can see there is a tuft of flagella at the ends. So on both ends you have tuft of flagella is present, bunch of flagella is present on the both ends. Okay, so here you see this is a vibrio. So it is a comma shape bacteria, that is called as a vibrio. So here vibrio, it is a comma shape. So like this there are 4 different shapes of bacteria present. First one is called as a coccus shaped. In coccus shaped bacteria, how they appear as spherical forms. So you can see these are the spherical forms. So these are spherical forms. Then if you see the bacillus, rod shaped, so you can see these are the rod shaped. So similarly the spirillum, so it is a spiral shape, the spirillum spiral shape, and vibrio, that is a comma shape. So like that there are different shapes of the bacteria. Now let us see about the structure of bacteria in detail. Actually let us see about the structure of bacteria. So this is the typical structure of bacteria, students. So if you clearly see that this is the typical structure of the bacteria. Okay, so this is the typical structure of the bacteria if you see clearly. So if you clearly see this bacteria has, you can see that in the cytoplasm. First let us see in the cytoplasm contents. If you see in the cytoplasm, here you can see there is a nucleoid is present. So nucleoid. So this is a nucleoid. It is not a true nucleus. So that is the reason it is called as a nucleoid. So it is a nucleoid. So the bacteria, the nucleus of the bacteria is called as a nucleoid. So why it is called nucleoid is because we don't have the true nucleus actually. The true nucleus is absent. So true nucleus is absent. Okay. So that's why it is called as a nucleoid. True nucleus is absent, it is called as a nucleoid. Next, so next you see here, so there is a outer substance, there is a sticky like substance that is called as a mucilage. Mucilage is present. Okay, so anyway, let us see the first cytoplasmic contents. Anyhow, so nucleoid, this part is called as a nucleoid. Okay, so if you see the nucleoid, so how this nucleoid appears, nucleoid is containing the DNA, no doubt about it, the nucleoid contains DNA. So if you see the type of DNA, it is it is a dsDNA. So dsDNA means the nucleoid is made up of double strand DNA. So it is circular, so nucleoid is circular closed, it is circular closed double strand DNA. Okay, so nucleoid of the bacteria. So if you see the cytoplasmic contents, first let us see about cytoplasmic contents. So if you see the cytoplasmic contents of this particular bacteria, if you see clearly, so if you see the cytoplasmic contents, so first the nucleoid, if you see, the nucleoid is nothing but it is made up of DNA. So nucleoid contains what type of DNA? It is having double strand DNA. And how the double strand DNA appears? It is circular and closed. The DNA is circular and closed. So double strand DNA is present, it is circular and closed. Then you see in the cytoplasm there is ribosomes are present. So ribosomes are attached to a thread-like structure. Here you can see. So this thread-like structure. So altogether is called as a polyribosomes. So what is this polyribosomes is composed of? So in prokaryotes, no doubt ribosomes are present. So ribosomes are in association with mRNA. So ribosomes plus mRNA. So ribosomes plus mRNA is called as a polyribosomes. So the type of ribosomes are, in case of the prokaryotes like bacteria, ribosomes are present and the type of ribosomes are called as a polyribosomes. So what is this polyribosomes? Polyribosomes are nothing but they are made up of ribosomes plus mRNA. So in the cytoplasm of the bacteria, polyribosomes are present. So ribosomes plus mRNA is present. So Fatima was asking me one question. So cholera is caused by Vibrio cholera. Yes, very good. So Vibrio is the bacteria, Vibrio cholera. So Vibrio cholera. So Vibrio is the genus, cholera is the species. So Vibrio cholera species causes the disease that is called as a cholera. It is correct. So once again, good evening students. So good evening, welcome to Nageshwar Rao and Fatima also. So let us continue with our topic. So ribosomes. So what type of ribosomes? They are polyribosomes actually. So what is polyribosomes are made up of? Polyribosomes are made up of ribosomes plus mRNA. Okay, ribosomes plus mRNA is called as a polyribosomes. Then, so let us see some more cytoplasmic contents. So apart from that, you have a small amount of RNA is also present. You can see very small amount of RNA, very small amount, not too much. And the storage bodies are present. So the storage bodies are called as the lipid globules. So lipid globules are present, storage bodies are present in the cytoplasm. Then, so if you see there is one specific structure, so that is called as a mesosome. So you can see here mesosome. So this mesosome is extension or originated from the plasma membrane. So mesosomes are originated from the plasma membrane, if you see clearly. So this is the mesosome. So mesosomes, they are invaginations. They are formed from the plasma membrane. So they are invaginations of the plasma membrane produces the mesosome. So what is the importance of this mesosome? So mesosomes plays a very much important role actually. So in case of plant cell or in animal cell, respiration takes place inside the mitochondria. But in case of the prokaryotic cell like bacteria, what happens is mitochondria is absent. So if mitochondria is absent, then how the bacteria will undergo respiration? So respiration. So mesosomes plays an important role in the respiration. So that is a very important point you have to remember. So mesosomes. So from where mesosomes are originated? So mesosomes are originated from the cell membrane or plasma membrane. The invaginations. So you can see in the diagram which I have drawn here. The invagination of the plasma membrane or cell membrane, the invagination of plasma membrane or cell membrane leads to formation of the mesosomes. Then what is the function of mesosomes? Mesosomes help in the respiration process. In plant cell and animal cell, what happens is respiration takes place with help of mitochondria, but mitochondria is absent in bacteria. So then how the respiration takes place in bacteria? And respiration takes place in bacteria with help of the mesosomes. So apart from that, there is another important function of mesosomes. Mesosomes also plays an important role in the binary fission. So mesosomes also plays an important role in binary fission. So mesosomes also plays an important role in the binary fission, so cell division. Okay, so how it is playing an important role? So it forms a septa or wall-like structure. During the cell division, it forms a septa. Septa is formed. It forms a septa. Okay. So during cell division, during binary fission, what happens is a wall-like structure will be formed. That wall-like structure later separates the two daughter bacteria cells during binary fission. So anyhow, so most important point you have to remember is the mesosomes plays an important role in respiration. Mesosomes plays an important role in the binary fission. So mesosomes plays an important role in the formation of a wall-like structure that is called as a septa. Okay. So inside the mesosomes, what happens is there are some enzymes. So inside the mesosomes, how the respiration is taking place means the enzymes required for respiration. So enzymes required for respiration. So enzymes required for respiration are present inside the mesosomes. So like that we have to remember some functions of mesosomes. Generally they ask in multiple choice questions. So what is the importance of this mesosome? So these are the important functions of the mesosomes actually. So if you see some more things about the, so that is about the cytoplasmic contents. Let us see about the cell wall. You see here cell wall is present. Okay. So cell wall. So cell wall is made up of peptidoglycan. We will discuss in detail anyhow. So you can see there is a this one. So this one, this part is called as a flagella. So this part which is coming out, this is called as a flagella. Okay. So you can see pilus, a short tube-like structure that is protruding out of the cell wall, that is called as a pilus. Okay, or sex pilus. So what is the importance of pilus? It is involved in the process of conjugation. It is involved in the process of sexual reproduction of bacteria, and that process is called as a conjugation. It is involved in the sexual reproduction of bacteria, and that process is called as a conjugation. So with help of sex pilus, the bacteria will attach to another bacteria and involved in the sexual reproduction of the bacteria, and the sexual reproduction of bacteria is called as a conjugation. So conjugation is the mode of sexual reproduction in bacteria. Generally bacteria undergoes asexual reproduction like binary fission, but bacteria also undergoes sexual reproduction through a process. That process is called as conjugation. Then how bacteria is undergoing conjugation? With help of a specific structure that is called as a sex pillae, or it is called pilus. So like that sex pilus is also present. So let us see in detail, students. Let us see in detail about the structure. This is the outline. So if you clearly see, when compared with the eukaryotic cell, this prokaryotic cell, so the common cell organelles, which is the common cell organelle present in both prokaryote and eukaryote. If you see the common cell organelle, so the cell organelle that is present in prokaryote is also present in the eukaryotic. Then there is only only cell organelle that is a common. So that is common for both prokaryote and eukaryote is ribosomes. So ribosomes are present in prokaryotic cells. They are also present in the eukaryotic cell. So no doubt about it. So welcome to few more students. Welcome to Rajita. So good evening, Rajita. So let us continue with the structure, students. So bacterial cell. So bacterial cell is covered by an outer slimy layer that is called as a capsule. So if you see here, the bacteria cell, so it is covered by a slimy layer that is called mucilage. You can see a slimy layer. So now this one is called as this mucilage layer. This is called as a capsule. So this outer layer is called as a capsule, that is covered by outer layer, that is called capsule. And middle cell wall, it is also having the middle cell wall is present. Then and inner membrane. So that inner membrane is nothing but it is plasma membrane. That inner membrane is nothing but it is the plasma membrane. So if you see the bacteria cell, so bacteria cell is covered by outer slime layer, that is or it is called as a capsule, and middle cell wall. So middle cell wall is present, and inner side is plasma membrane. You can see here there is plasma membrane is also present. Here you can see plasma membrane. So this part. So this part is plasma membrane. This part. this part is cell wall, okay, and this part is the slime layer. So 3 layers here. So this one is slime layer, mucilage slime layer or capsule it can be, and this part, central part is cell wall, and at the last, so that is plasma membrane. You can see clear here, you can see plasma membrane. This part is plasma membrane. So bacteria outer covering, so bacteria is covered by a slime layer, cell wall and inner membrane. Let us see. So cell wall, let us see about the slime layer or capsule. So cell wall of most bacteria occurs by a viscous gelatinous loose sheet. So bacteria is covered by a viscous gelatinous loose sheet. So that slime layer is a loose sheet actually, and it is a gelatinous. So gelatinous or viscous, it is gelatinous and viscous. Okay. So how the slime layer appears, if you take the bacteria and observe under microscope, so that mucus layer is nothing but it is gelatinous or viscous in nature. So that layer is called as a slime layer. So when these components of layer are compactly arranged, if the layers are compactly arranged or tightly arranged, then it is called as a capsule. If that outer layer is a loosely arranged, then it is called slime layer. If the tightly or compactly arranged, it is called as a capsule. So what is the function of the capsule? If you see, the capsule serves as a protective layer. So main function of the capsule is it is acting as a protective layer. So generally the bacteria which is having capsule will not have flagella. So that's what he was telling here. So bacteria, the capsulated bacteria usually don't have flagella. So that is an important point which we have to remember here. Now let us see about the cell wall. So cell wall is the most important thing which we have to remember. Generally they ask multiple choice questions only on the cell wall. So what is cell wall of bacteria is made up of? You see, cell wall of bacteria is made up of polysaccharides. So in multiple choice question they will ask you what is cell wall of bacteria is made up of. Simply in 2 terms, you have to remember the cell wall of bacteria is made up of sugars plus amino acids. This is the 2 terms you have to remember actually. So what is cell wall of bacteria is made up of? Cell wall of bacteria is made up of sugars plus amino acids. So cell wall of bacteria is made up of sugars plus amino acids. That sugars are nothing but what type of sugars it is? They are polysaccharides. And what are the names of that polysaccharides? The names of the polysaccharides are NAG and NAM. So what is full form of NAG? If you see the full form of NAG, they are anestyl glucose amine. So NAG full form is anestyl glucose amine. So okay. So NAM. So what is NAM? Anestyl muramic acid. So what is a bacteria cell wall is made up of? In multiple choice question they'll ask you. In 2 terms, in 2 terms you should mention like they are made up of sugars plus amino acids. And what type of sugars they are? They are polysaccharides. And what are the polysaccharides? Those polysaccharides, which are those polysaccharides? They are NAM and NAG. What is the NAM and what is NAG? So if you see the NAG, so what is full form of NAG? Anestyl glucose amine. What is the NAM? So anestyl muramic acid. So this NAM and NAG both are polysaccharides. They are the polysaccharides. So they are polysaccharides. So just now we were discussing, right? So made up of polysaccharides. They are polysaccharides. So we already know that polysaccharides are nothing but they are sugars. So like that, so bacterial cell wall is made up of sugars plus amino acids. Now what are those amino acids it is made up of? If you see the amino acids it is made up of amino acids D-alanine, so L-alanine, D-glutamic acid and L-lysine. So it is made up of amino acids like D-alanine, L-alanine, D-glutamic acid and L-lysine. So these are the things it is made up of. So it is made up of amino acids like D-alanine, L-alanine, so D-glutamic acids and L-lysine. So that is importance of the cell wall. So once again, so welcome to few more students. So welcome to Santu. So if you see the cell wall once again, students, cell wall is the most important topic which you have to remember here actually. So cell wall of bacteria is made up of sugars plus amino acids. It is made up of sugars plus amino acids. Okay, no doubt about it. So what are the sugars that are present in the cell wall? The sugars are nothing but they are polysaccharides. Okay, what are those polysaccharides again? The polysaccharides are known as NAM and NAG. So what is NAM and what is NAG? So NAM is anestyl muramic acid. So what is NAG? NAG is anestyl glucose amine. So this NAM and NAG, if you see this NAM and NAG, so NAM and NAG, so these are joined by glycosidic bond. So the NAM and NAG are also joined by a specific bond that is called as glycosidic bond. So actually NAM and NAG both are sugars, Generally two sugars are joined by glycosidic bond. So as usual here also